Okay, sorry there. I was having some issues. So, I apologize. I need to have the fan running because it's kind of toasty in here. It's kind of hard for my HVAC to keep up. So, anyways, we are going to continue this review on the testis in immune privilege, and we had recently gone over the physiology, or the physiology of the testis, and what cells are there, what cells are present in the testis, and how there is both a physical barrier between the developing sperm and the immune system, which helps confer uh, resilience to the sperm cells, but then also there has to be some hormonal or cytokine level um, suppression of the immune system in the testes, because there is the ability for a lot of tissues to be grafted into the testes, even exposed, even on the other side of the blood testis barrier, which ends up, which would one would normally assume that would cause some immune response, some attack on that foreign tissue, but even there, the immune system is suppressed in some way to allow that foreign tissue to survive and exist. So we are going to switch the our focus from the actual physiology to the hormonal level, or to the immunological level, rather than the testes level. So uh, let's begin. Role of leukocytes in maintenance and disturbance of testicular immune privilege. So macrophages. In the adult testis of most species, mononuclear phagocytes, monocytes and macrophages, um, represent a substantial cellular population of the interstitial compartment. So remember, the interstitial compartment was this area of the testes. So here you have your seminiferous tubules over here, and then here you have your interstitial compartment that has a lot of the white blood cells. So this area is more exposed to the immune system, right? Anyways, in rats and mice, by far the two best studied species, the ratio of testicular macrophages to Leydig cells is about 1 macrophage to 4 or 5 Leydig cells. Under normal conditions, macrophages and other leukocytes are exclusively found in the interstitial space. So remember, these here you have your blood testis barriers over here, and you have all these tight junctions within the Sertoli cells that protect the developing sperm and germ cells from the immune system. Well, here you have a bunch of, if you're on this side, this is where you are going to find the immune cells. And they're also saying that for every 5 Leydig cells, wait, let me, yeah, I should have said these are Sertoli cells. If I misspoke, sorry. But here you have your Leydig cells, and for every 4 or 5 Leydig cells, you have 1 macrophage. Okay, so in humans, they are also found in the tubular wall, but never within the seminiferous epithelium. So in humans, they are also found very, very close to the seminiferous epithelium, but they are never within the seminiferous epithelium. Only under pathological conditions, or in the regressing testis of seasonal breeders, such as swan, can macrophages enter the germ cell compartment, where they phagocytose degenerating germ cells as the so-called spermatophages. So remember, phagy means eating. So a spermatophages would be something that is eating sperm, or consuming, surrounding it. So, or consuming it by surrounding it. So, in humans, though, you will not see this happening unless you are undergoing some sort of pathological condition. So, but in the testes of seasonal breeders, such as swans, you can see the macrophages going into where the germ cells are. But that is rare. And remember, humans, we breathe year round, right? We breathe all the time. So we don't undergo that same process. However, in apparently swans, and I wish that they listed more here, you can have this happen during the off season. So during the off season, there might be some evolutionary advantage to having the immune cells go up into your testes. Continuing, I should say go up into the seminiferous tubules because they are in the testes, but they're not in the seminiferous tubules normally. So in the testes of men with impaired spermatogenesis of differing etiology or different causes, the number of CD68 positive macrophages within the tubular wall has been found to be increased. So they're saying is in men with impaired sperm production, you can see a specific type of macrophage uh, entering into the seminiferous tubules. And the CD68 positive, I'll look this up, but this probably just means that 68 is probably the type of one of the receptors that they have, but we'll look it up. I should replace it with a positive. Yeah, so So CD68 cluster of differentiation is a protein highly expressed by cells in the monocyte lineage by circulating macrophages and by tissue macrophages. So this is a protein that is highly expressed, which is why they call it CD68 positive macrophages in the paper. So what is the function of the CD68? So it's a transmembrane, which means it goes through the cell membrane, and is heavily glycosylated in the extracellular domain. Okay, but what does it do to other? It's related to others, and then the scavenger receptor, which typically function to clear cellular debris, promote phagocytosis, and mediate the recruitment and activation of macrophages. Okay, so yeah, I won't be able to redigest and regurgitate that within a few seconds. So we will continue. Just know that this is, when it comes to a lot of immune cells, we often designate certain numbers with positives on them to tell the reader what proteins these immune cells are expressing. So CD68 positive means that these macrophages or these certain immune cells are expressing the CD68 protein. And we do that with a lot of other proteins, so it's not just CD68. So you will, when looking at immune cells, you will frequently see CD number and then positive or negative after that. Continuing, testicular macrophages not only possess all features common to macrophages at other sites, but also exhibit testis-specific functions. So we're saying here is that not all immune cells are the same. You have a distribution of immune cells throughout your entire body. And so some of the immune cells in the testes are not only similar to immune cells that you can find all over the body, but they also have additional functions associated with them. So regarded as essential for male reproductive function, macrophages display a particularly close morphological and functional link with the predominantly interstitial cell type, the Leydig cells, reflected by intimate specialized interdigitations. So I'm assuming interdigitations means it has a bunch of like finger-like things coming off of it, but I'm going to search that up just to make sure. Oh, and I forgot to mention at the beginning of this, this is not my field of study at all. So if I make mistakes, which is very likely, just leave them down in the comments. But here, interdigitations or digitations are, let's go to Wikipedia, because I'm more comfortable The interlinking that resembles the fingers of two hands being locked together. So that would probably... So yeah, I'll go with that, with Wiktionary, but... Okay, so here you can see the Leydig cells have, I guess, digitations, right? But what is this? This is the dendritic cells. Well, dendritic cells, I think, always have these dendrites or branches coming off of them. So yeah, this is throwing me for a loop a little bit. So let me read this again. Macrophages display a particularly close morphological and functional link with the predominant interstitial cell type, the Leydig cells. Because they're okay. So the predominant interstitial cell type is the Leydig cells. These are the cells that you find mostly in the interstitial space. So going back to this photo, you can see the most common cell that they have drawn here is the Leydig cells, right? And these are, as I said earlier, associated with the production of some hormones. So let's go back down. Regarded as essential for male reproductive function, macrophages display a particularly close morphological and functional link with the predominant interstitial cell type, the Leydig cells, reflected by the intimate specialized interdigitations. So when I read this, what I'm understanding is these macrophages have finger-like projections coming out of it, and the Leydig cells also have somewhat finger-like digitations or coming off of it, finger-like things, and they are somewhat linked together. But you know, I'm not a doctor, so this isn't my field. So if I'm misconstruing or misunderstanding this, please let me know. Moreover, they play an important role in Leydig cell development and the regulation of steroidogenesis in adults. So, okay, so this is something that we know. We know that the Leydig cells play an important role in the production of androgens, in the production of different hormone cells. But it's not just the Leydig cells. Nothing exists in a vacuum, right? No cells exist in a vacuum. So the immune system of the testes is specialized to work with the Leydig cells in order to produce hormones. Continuing, testicular macrophage numbers are maintained at comparatively high levels through a direct Leydig cell-mediated mechanism rather than any influence of the seminiferous tubules. Interesting. So the macrophages, which is a subset of the immune cells, they are maintained at high levels in the testes through the Leydig cells. So the Leydig or the Leydig cells are participating in a way to keep those immune cells high, which could concern me, right? It could concern me knowing that if I have a dysregulation of my immune system in my testicles, since there is a large number of the immune cells, and since they play such a critical role in the development of my hormones, then that probably would indicate that my hormones will get screwed up if I get inflamed in the testes, right? So that's good to know. With testosterone and macrophage migration inhibitory factor, or MIA, playing only a minor role, if any, in this regulation. So interesting. So testosterone and macrophage or MIF are playing a small role. So this isn't just in the testes. The migration inhibitory factor is something that I believe would prevent macrophages from moving around, right? So let's go to Wikipedia and let's see if we can gain insight within a few seconds. So known as glycosylation inhibiting factor, or this is a protein in humans that is encoded by this gene, and it's an important regulator of innate immunity. The MIF protein superfamily also includes this. Bacterial antigens stimulate white blood cells to release MIF into the bloodstream. And then this binds to CD74. So remember, this would be another type of protein that immune cells can express. And if you were to read a CD74 plus immune cell, you would therefore know that it is expressing the CD74 receptor, right? So this receptor is on a subset of the immune cells. And then when this MIF, or the macrophage migration inhibitory factor, is released and circulating in the blood, it will bind to this CD74 and trigger an acute immune response. So it'll make them go crazy and think that there's something nearby. So MIF is classified as an inflammatory cytokine. Furthermore, glucocorticosteroids also stimulate white blood cells to release it. Okay, so, and trauma activates the pituitary gland to release MIF. Okay, so let me just read this real quick just to see if there's anything that I should mention. MIF also contains cysteine, alanine, leucine, cysteine, catalytic sites. Okay, I don't think there's anything that is worth going over too deeply. I think we can be a little bit superficial with that. So anyways, but that would normally often play a role in macrophage. This MIF would normally play a large role, but in the testicles, it doesn't seem that the macrophage numbers are affected by MIF, which would be surprising, which means that the Leydig cells are using a completely different mechanism to keep those macrophage numbers high. Again, in Leydig cell depleted testes, only 50% of the normal macrophage population persists. So if you remove the Leydig cells, you will lose half of the macrophages. So the Leydig cells are purposefully keeping macrophages there. Besides their well-established interactions with Leydig cells, testicular macrophages also influence Sertoli cell function and spermatogenesis by releasing soluble mediators. An example, macrophage activation enhances a human chorionic gonadotropin-induced disruption of spermatogenesis in the rat, possibly by increasing sensitivity to interleukin-1. So I won't be able to digest that. Just, I guess what I'm understanding from this is that the macrophages are screwing with the hormones in the area, but I wouldn't be able to. Wait, besides what else? So these macrophages are releasing some sort of a nearby soluble hormone, which is able to affect spermatogenesis. But yeah, this is above my my understanding. Continuing, there is little doubt that macrophages play a central role in the establishment and maintenance of the immune privilege of the testis. This supposition was first substantiated by in vitro studies where testicular macrophages displayed a reduced capacity to synthesize interleukin-1 beta and TNF alpha, which something nuclear factor alpha, tumor necrosis factor alpha, and it's an inflammatory cytokine. So these macrophages that are highly produced or that are induced to exist in the testes, these have a reduced capacity to synthesize interleukin beta and this tumor necrosis factor alpha. And this one is an inflammatory cytokine, and this is, I would assume, also a pro-inflammatory cytokine, but I'll search it up just to make sure. Well, let's see. This cytokine is produced and activated by macrophages as a protein, which is proteolytically processed to its active form by caspase 1. So it contributes to inflammatory pain, hypersensitivity. Okay, so it is a pro-inflammatory cytokine. Um, com- Continuing, compared with macrophages from other tissues. So these macrophages in the testes are do have reduced abilities to generate these pro-inflammatory cytokines, which makes sense, and exhibit immunosuppressive characteristics. So if you're not secreting the pro-inflammatory cytokines, they are functioning as immunosuppressive in the testes. However, synthesis of granulocyte macrophage colony stimulating factor and prostaglandins E2 and F2 alpha was not inhibited in these studies. So it sounds like they are releasing other types of hormones that we associate with stimulating the immune system, if I remember correctly. Um, but I think they're just putting this here because this is something of note. Pointing to a heterogeneous macrophage population. Okay, so it's just saying that they have a variety of different macrophages, but I won't be able to tell you too much about that. In a different model, macrophage interleukin-1 production was initiated after endotoxin challenge, but as this production was accompanied by a corresponding decrease in the expression of Sertoli cell-derived interleukin-1 alpha, bioactive interleukin-1, levels in total testes remained unchanged. Oh, okay, so interesting. So using an endotoxin or using just a toxin to initiate the, or to, I would assume that this simulates the presence of some type of bacterium. The macrophages ended up actually producing interleukin 1, which, remember, they normally are suppressive, like they normally don't produce it as much, but in the presence of this endotoxin, they do produce it. But even when the macrophages produced it, then the Sertoli cells stopped producing the same chemical. So the total amount in the testes remained pretty much constant. So that's interesting. So there seems to be a mechanism going between the immune cells and the Leydig cells. Sorry, and the Sertoli cells, which are the nerve cells within the seminiferous tubules. There's some interplay going on that prevent that when interleukin alpha gets expressed by the white blood cells, then the Sertoli cells decide, okay, we're not going to produce this. Interesting. Furthermore, systemic lipopolysaccharides challenge in immature and mature testes resulted in enhanced levels of regulatory cytokines, interleukin-6, and constitutively elevated the production of the anti-inflammatory mediators activin A and TGF-beta in both normal and inflamed testes. So, systemic lipopolysaccharide challenge. I assume that this is, so lipo meaning fatty and polysaccharide meaning a carbohydrate, basically. So a carbohydrate that has the addition of fatty groups on it can apparently cause either damage or stimulate. It can challenge the testes in one way, shape, or form. So let me look this up just to make sure I have this right. Oh, I see. So by injecting lipopolysaccharides into tissues, you are in effect simulating the presence of bacteria. And when you do that, then the immune system sees those lipopolysaccharides and then starts acting as if there is bacteria in that location, right? And so if you are injecting the lipopolysaccharides in the immature and mature testes, it results in enhanced levels of the cytokine interleukin 6 and will forever elevate the production of the anti-inflammatory mediators activin A and this TGF beta. And it was irrespective of whether or not the testes were inflamed or not. Interestingly, TGF beta 1 and interleukin 1 beta were mainly present in their latent inactive forms, which suggests multiple levels of regulation in the testes-specific manner. There are too many positives and negatives, and I apologize. I can't go over all this to digest it. If you understand it, you can leave it down in the comments. But they're just saying that there's multiple methods of regulation. So even though they're seeing the expression of some of these cytokines and some of these hormones, sometimes they're in the inactive form versus the active form. So just know that there are multiple levels of regulation. Anyways, the diminished capacity for expression of inflammatory cytokines such as interleukin 1 beta and TNF alpha by testicular macrophages, the testes is capable of mounting an inflammatory response. The diminished capacity for expression of inflammatory cytokines such as or testicular macrophages, the testes is capable of mounting an inflammatory response. Oh, okay. Sorry, I was missing the first three words. So even though there is a diminished capacity that that it is harder for them to do it, there is still the possibility for these immune cells to do it if the need really, really arose. Okay, that makes sense. There is now increasing evidence that the seemingly contradictory situation is based on substantial contribution of the testicular somatic cells to the inflammatory response by producing various pro-inflammatory mediators such as MIF, inducible nitric oxide synthase, and interleukin-1 isoforms. Okay, so they're saying here that since we are able to express or find ways to simulate infection, the immune cells are still doing some types of releases of hormones that are associated with inflammation. They are trying to inflame the area. But it appears as though that the mediators of this inflammatory response are coming from the Leydig cells, Sertoli cells, and the peritubular cells. So whatever cells are in the testicles, those are playing an active role in suppressing the immune response of the testes, even though the immune cells themselves also have impaired abilities in of themselves. We can move around those impaired abilities by just overloading the white blood cells with pro-inflammatory signals. But even then, a lot of their releases of cytokines are inhibited or screwed up by the Leydig cells, Sertoli cells, and the peritubular cells of the testes. So this harkens to multiple strategies are being employed in order to suppress the inflammatory response of the testicles. So that's what I'm getting out of this. In the rat testes, at least two subsets of macrophages can be discerned. One which can be differentiated by its expression of an approximately 100 kilodalton lysosomal glycoprotein. So a lysosomal glycoprotein would be a protein that is glycosylated, so it has sugars on it, which would indicate that it's probably secreted or at least expressed on the surface of the cell. And it's lysosomal, which is something that would cause the lysis of organisms. So it would find a way to disrupt the cellular membrane of a creature or a cell and basically destroy it so that its contents spill out into the environment. Anyways, in the rat testes, at least two subsets of macrophages can be discerned, one which can be differentiated by its expression of this lysosomal glycoprotein. And this glycoprotein is recognized by the monoclonal antibody ED1, which is a rat homologue of human CD63. Okay. Sorry, I'm just trying to make sense of this. So, and the other more common variety, which is a surface antigen recognized by the monoclonal antibody ED2, which has a human homologue CD163. So I've seen CD163 before, but okay. I think I'm getting this. So in rat testes, there are two subsets of macrophages, and these two can be differentiated based on whether or not they have a protein that is recognized by ED1, or there's another protein that is recognized by ED2. And they're saying that these two rat macrophages are associated or have homology between the human CD63 and the human CD163 macrophages, which I think this is it the CD163 macrophages that are ACE2 positive. Let me search this. Furthermore, overexpression. I'm gonna just look through this real quick, just to see. So CD163. Yeah, so I think the CD163 positive immune cells might be ACE2 positive. Let me just read this real quick. So that the upregulates. Wait, find ACE2. Others report that Adam 17. Okay, this isn't a good one. Maybe this paper. Moreover, the epithelial cells lining the salivary ducts. Collectively, these findings demonstrate that a broad range of people. Okay, so maybe it wasn't. I'll let you know, because I do know that there is a type of immune cell that is ACE2 positive, but I can't remember what immune cell it was explicitly, because so yeah, just look at it. This is why immunology and biochemistry is so unapproachable. Look at the MAC387 positive, the CD163 positive, the CD68 positive, the MAC. Well, yeah, I said that already, but like, there's so many proteins that you got to keep straight that it makes it difficult to really make sense out of any of this. But yeah, we'll keep on going. Just just bear with me, right? I'm sorry that we went on a tangent uh, without a punchline, but that is the way that this goes. So, anyways, continuing. While the ED2 positive resident type subset forms the majority of the macrophages in rat testes, a significant portion of the ED1 positive and ED2 negative cells is also present, which is about 15 to 20%. Which, remember, this is what we were talking about over here. You have some that are ED1 positive, and you have some that are ED2 positive, and those are the main two subtypes of macrophages in the rat testes. Presumably representing circulating inflammatory monocytes or recently arrived macrophages. This heterogeneity has functional implications as in the testes ED1 positive subsets, but only few ED2 positive resident macrophages express MCP1 and I nitric oxide synthase, I think is what that is. So yeah, so nitric oxide synthase in untreated and lipopolysaccharide challenged rats. So let me read this real quick. This heterogeneity has functional implications, as in the testes ED1 positive subsets, but only few ED2 positive resident macrophages express the MCP1 and the nitric oxide synthase in untreated LPS challenged rats. So I think they're saying that these ED2 positive ones might just be from chance, but man, I have such a hard time keeping track. Regardless, a possible third and yet uncharacterized small subset has been indicated based on its ability to synthesize interleukin-1 beta. Furthermore, a subpopulation of mouse testicular macrophages, isolated by density centrifugation, has been found that express high levels of the TGF-beta and exhibits a tolerogenic phenotype. So, furthermore, a subpopulation of mouse testicular macrophages, isolated by centrifugation, can express high levels of the TGF-beta. And this high expression of TGF-beta, I assume, is contributing to the tolerogenic phenotype, or the ability for the testes to tolerate antigens. The immunosuppressive qualities of this subpopulation were recognized by its inability to induce T lymphocyte proliferation. So T lymphocyte proliferation would be when the T lymphocytes get a signal, and the T lymphocytes are a different type of immune cell. If they get a signal in an area to proliferate, that means that they are charged to attack whatever's in the area, and so they're duplicating in order to fight whatever infection is in that area. So I'm just trying to see if I should go into that, or if that's a good enough explanation. I think that's a good enough explanation. So a feature abrogated after adding neutralizing TGF-beta antibodies and its reduced antigen presenting activity. So this feature of having basically the inability to induce T lymphocyte proliferations by these small subs. Geez, I can't. Recently, third as yet uncharacteristic small subset has been indicated based on. So yeah, by the small subset, has been indicated based on its ability to synthesize IL-1 beta. And then there's another subpopulation isolated by centrifugation that has been found to express high levels of this TGF-beta, and it exhibits tolerogenic phenotype. And the immunosuppressive qualities of this subpopulation were recognized. We could figure this out because it wasn't able to induce T lymphocyte proliferation. But if you add these TGF-beta antibodies, or antibodies that will sequester these TGF-beta hormones, then the feature, or the immunosuppression and the inability to induce T lymphocytes was lessened. So that means you could end up inducing T lymphocyte proliferation if you got rid of the TGF-betas. Of note, in an unseparated preparation of total testicular mouse macrophages, this immunosuppressive activity prevailed. So if you didn't bother separating these macrophages from each other. So what they're doing is they are separating these macrophages through centrifugation or other methods. They might also use things like flow cytometry to do this. But if you can separate all these macrophages and find out what their qualities are in immune regulation in of themselves, because remember, biology is a noisy field. So if you can remove the noise, then you can understand the individual components. Now, if they decide not to do any of the centrifugation, if they decide not to do any of the separation, in an unseparated preparation, this immunosuppressive activity prevailed. So if you separate everything, you can find a subset of the white blood cells that are contributing to the immunosuppressive effects. And if you don't do any centrifugation, you can still see those immunosuppressive effects. Man, that was a little complicated. God. Jesus, this is long. I might have to do a third video. These observations provide further support to the concept that at least two macrophage subsets exist in the adult testes that differ in the expression of markers and inflammatory mediators. In the rat, the ED2 positive resident population of testicular macrophages does not participate in promoting inflammatory process. It is believed to have an immunoregulatory role in maintaining immune privilege and tropic function, particularly on Leydig cells. Clear evidence points out that the ED1 positive, ED2 negative monocytes and macrophages are involved in the testicular inflammatory response, and it is the influx of ED1 positive monocytes during acute and chronic inflammation that drastically altered the composition of the macrophage population. Okay, so these ED1, let me just read this. Yeah, so they're saying that the ED2 positive cells, so let me look just to make sure. Oh, I went way too high. So the ED2 positive ones are the ones that we find in the testes usually. These are the resident ones, right? And then these ED1 ones are also just there, but they are there in a smaller proportion. They're like between 15 and 20%, whereas the ED2 positive ones are 80 to 85%, right? And then these ED2 positive ones tend to be immunosuppressive. They tend to be incapable of initiating the immune response as well. But you can have an influx of these ED1 positive monocytes during acute and chronic inflammation, and this will drastically alter the composition of the macrophage population. So, okay. So now it's starting to make sense to me what they're trying to get. Normally you have a vast majority of the macrophage population in the rat testes participating in a very anti-inflammatory, I should say, participating in creating an anti-inflammatory environment, right? But in chronic and acute inflammation, you need to have the influx of these ED1 positive, ED2 negative immune cells that are normally in low numbers in the testes. You need to get an influx of those in order to create the more of an inflammatory response. So this could tell you that if you do have inflammation of your testicles, it wasn't just an willy-dilly, or it wasn't just your testicles are getting inflamed. It is your testicles lost its anti-inflammatory ability and then got inflamed. So if your testicles are inflamed, you need to be worried about that, because then the all those protective measures have been overridden. They have been overridden, and that could tell you that something is seriously wrong. Interesting. Okay. So, and shift the cytokine balance in favor of an inflammatory response with the potential to overcome the immune privilege. So this is something that is also worrying, because if you ever move into having a pro-inflammatory response, it's not just going to be attacking the bacteria. It is also therefore exposing your own sperm cells to your immune system, right? So if your balls get inflamed, it is not just the disease that you need to worry about, that the immune system is fighting. It is also the effects of having your sperm be exposed to your immune system. And that can have very drastic consequences on your hormone production or your sperm production. Okay, continuing. It is noteworthy to say that the testis, at least in acute inflammatory models, involving single injection of lipopolysaccharides, appears to possess a robust mechanism to counterbalance the influx of new ED1 positive monocytes, as the increase is only temporary and resolved after 1 to 2 days. So if you give an injection of the lipopolysaccharides to convince your immune system that the balls are infected, you will have an influx of these ED1 positive monocytes from elsewhere in the body. They come in because they're pro-inflammatory when the immune cells of your testes are normally anti-inflammatory. These pro-inflammatory ones come in, they do their thing, and then very quickly your testicles are trying to maintain that anti-inflammatory environment, so they quickly try to remove those macrophages, the ED1 positive macrophages. This mechanism is clearly not the case in chronic testicular inflammation such as autoimmune orchitis, where a strong, lasting increase in macrophage numbers is characteristic. So remember, this is when we do the LPS injection, which is just, it's an acute thing. It's an acute thing to inject lipopolysaccharides into the testes. But if you have an autoimmune response, or you have the experimental autoimmune orchiditis, or oh gosh, I can't. Experimental autoimmune, inflammation of the balls, then this is being completely overrun, and this autoimmune response is having a lasting effect on the testes. So whatever mechanism that is used to remove these pro-inflammatory white blood cells, that is thrown to the wayside later in chronic cases, which is unfortunate. So obviously, other yet unknown mechanisms are in effect to override the protective mechanism, probably involving T lymphocytes and dendritic cells, with detrimental consequences for testicular function. So if you have chronic inflammation of the balls, there is a detrimental consequence for testicular function. So several important questions arise from these data. What are the local factors that recruit, then resolve ED1 positive monocytes to form in the testes in acute inflammation. So what is recruiting these white blood cells to come in, and then what is telling them to get the hell out when the inflammation should be done? We don't know this yet. And what causes the chronicity of elevated macrophage numbers in autoimmune orchitis? Which local factors maintain the balance of ED1 negative and ED2 positive resident macrophages and ED1 positive, ED2 negative inflammatory monocytes, macrophages in the testis under normal conditions? While the mechanism that controls the resident testicular macrophage phenotype is still unknown, evidence increasingly points to the influence of testicular somatic cells. Leydig cells, Sertoli, and peritubular cells are all known to produce a plethora of cytokines. So these somatic cells of the testes are producing a lot of cytokines, which could very easily affect what type of immune cell populations you have. So although we do not know the mechanisms by which the testicular macrophage populations are being altered, it is probably, the evidence is pointing towards these somatic cells. So, and it can be speculated that these cells act in concert to first recruit, and then, depending on their localization and hence the cytokine milieu, it dictates macrophage type and function. This hypothesis presupposes that the somatic and immune cells of the testes act together to provide an environment that protects the germ cells from autoimmune attacks. So the whole theory, the whole supposition, the whole hypothesis is that your body is going through all this work explicitly for the germ cells, to protect the germ cells from autoimmune attack. There could be another function that we don't know about, but this protection of the germ cells is what we have to go by, and it makes the most sense, because if it screws up, you can see that the germ cells are getting damaged, right? There could be another reason, but most likely it's for the protection of the germ cells. In this concert, TGF-beta and possibly activin A appear to play a critical role by inhibiting specific immune responses, finally minimizing the risk of autoimmune reaction to testicular self-antigens, and therefore maintaining immune privilege. So they're just saying the TGF-beta and the activin A appear to be the critical components that are causing this anti-inflammatory and immunoprivileged status. The recent study of these people shows, however, that in addition to anti-inflammatory or regulatory cytokines, other classes of molecules attribute to our understanding of the immune privilege of the testes. They isolated lysoglycerophosphatidylcholines, specifically the C16 saturated and C18 unsaturated ester-linked lysophosphatidylcholines from rat interstitial fluid and bovine follicular fluid. These compounds showed a strong T lymphocyte inhibition in vitro and can provide a rationale for the immunosuppressive activity of gonadal function. So these lysoglycerophosphatidylcholines and these lysophosphatidylcholines, these are amphipathic molecules, sort of like soap, but they function as they are functioning as hormones that have been seen in the rat interstitial fluid and the bovine follicular fluid, and they appear to also be showing a strong T lymphocyte inhibition, so slowing down the immune response, suppressing the immune response. So it appears to not just be these cytokines that we need that are mediating this response. It appears to also be these other simpler molecules. These doesn't sound simpler, but the lysoglycerophosphatidylcholines and the lysophosphatidylcholine's. These also appear to be playing a role, but you don't need to memorize that. So that was just the macrophages. And now we are moving to a different cell type, the different immune cell type, the dendritic cells. So dendritic cells, or DCs, are a heterogeneous population that belong to the most important antigen-presenting cells. So antigen-presenting cells are immune cells that are presenting different types of antigens on their surfaces. And this would be a way to determine if an immune cell should survive or not, right? And they play a major role in the initiation and orchestration of primary immune response of both the helper and cytotoxic T and B lymphocytes, the effector cells of the adaptive immune system. So these dendritic cells play a major role at presenting antigens on their surfaces and are what tell the cytotoxic T and the B lymphocyte cells what they should go after, if that makes sense. Continuing, dendritic cells not only activate lymphocytes, but also tolerize T cells to antigens, so thereby minimizing autoaggressive immune response. So these dendritic cells can present antigens that your own body produces, and they do it in a way which tell the other immune cells, don't attack these antigens because these antigens are a part of you. And I thought it did that by killing immune cells that ended up attacking those antigens, but I could be mistaken or misremembering. Anyways, they arise from the CD34 positive bone marrow progenitor cells, or CD14 positive monocytes, and differentiate into at least 3 distinct subsets: the Langerhans cells, the interstitial dendritic cells, and the plasmacytoid dendritic cells. So in the bone marrow, I think, is this the receptor that HIV goes into? HIV receptor. Oh, it's a CD4 receptor. Okay, never mind. Sorry, false alarm. So the CD4 or CD34 positive, this is a protein that is expressed on the surfaces of some cells in the bone marrow. And they're important because some of them will differentiate or turn into these dendritic cells, which play a very important role in teaching the immune cells what to attack and what to not attack, right? They're just saying where they're coming from. So they come from the bone marrow or the CD14 positive monocytes. Anyways, the dendritic cells migrate as immature or precursor cells from the bone marrow into peripheral tissue, or into different tissues, where upon receiving an activatory signal associated with pathogens or inflammation, they migrate to the local lymph nodes, mature, and present the antigen to T cells captured in the periphery. So these dendritic cells get a signal associated with pathogens. So they might go over this later, but it could be something that the pathogens secrete themselves, or it could be something that your innate immune response ends up releasing in response to these pathogens. I'm brushing up right now. Anyways, after they receive that signal, they go into a local lymph node. They'll go to a nearby lymph node, and they mature in that lymph node, and then during the maturation process, they start presenting the antigen. Yeah, they start presenting antigens on the surface, which can tell the immune system what to attack later. And they present the antigens to T cells captured in the periphery. Okay. This somewhat simplistic model of the life cycle of the dendritic cells has been corrected in recent years to accommodate the heterogeneity of dendritic cells found in vivo. Although it is clear that not all dendritic cell types follow this life cycle precisely, as a generalized model, it is still a valuable tool for illustrating what occurs during the initiation of immune responses. So this is one of the reasons why it is so difficult to teach and talk about what the immune system does, is because, biology is noisy. Biology is really noisy. There is a plethora of different proteins that you got to keep track of that are expressed in different cells, and so they say, oh yeah, the CD14 or the CD163 positive, like it's all just noise, right? It's all really complex and hard to follow. But frequently we simplify we have a simplified model, right? So I forgot who said it, but all models are wrong, but some are useful. And in this case, we have a wrong model, but it's a useful model. It might not have everything, it might not consider every single variable, but it is sufficient to teach to students, and it is sufficient to understand, okay, what is the most important thing that these cells do. And maybe down the road we discover more and we say, oh yeah, there's a completely other response or function that these cells are doing, and that is actually the main role. But right now, this is all that we have, right? So the only thing that refutes science is better science, right? So if we discover more, we might be able to explain more. Anyways, immature dendritic cells have the highest capacity to internalize antigens, but low T cell stimulatory activity. Whereas mature dendritic cells downregulate their endocytic activity and are excellent T lymphocyte regulators. So what does this tell me? This tells me that the young dendritic cells are able to, they can grab a lot of things in their environment and then present those out on their cell surfaces to warn the other immune cells what to do. These dendritic cells have a great capacity to take new things, but you wouldn't want a dendritic cell to take the wrong thing, present it, and then that tells the T cells to attack something that it shouldn't attack. So the young ones can take a bunch of different stuff, but they are very difficult at convincing the T cells what to do. Makes sense, right? But the mature ones, the mature dendritic cells, they already took a lot of stuff, right? And probably focused their energy on the antigens that made most sense later in their life. So although they don't have the ability to change nearly as much, what they say should, or what they present should come with a lot of weight because they are mature. So as a result, they might not be as good at changing as easily, but they are, for lack of a better term, they're wise, right? They're wiser, they're more mature, and therefore what antigens they do present has probably withstood the test of time, and therefore the T cells get highly activated by what they have to say. So there's a lesson in there somewhere, but we will skip that and move on. Anyways, mature dendritic cells are characterized by the upregulation of surface T cell co-stimulatory and major histocompatibility complex class II molecules. The production of bioactive interleukins 12 and TNF-alpha and changes in migratory behavior. So the way that mature T cells, or sorry, mature dendritic cells, the way that they convince the T cells to listen to them more is through the higher expression of co-stimulatory, so CD40, CD80, and CD86. So these are different types of membrane proteins that they can be expressing. And if you up-express them, these things that are on the surfaces of the dendritic cells help convince a T cell that, yes, whatever you say or whatever you're presenting is very important, right? Whereas an immature dendritic cell doesn't have though as many of those receptors on their surfaces. But those immature dendritic cells have the ability to experiment more. The immature dendritic cells are able to experiment and take more things in, and that's something that the mature T cells just don't. God, the mature dendritic cells they're a little bit more conservative. They can't take in nearly as much, and they're more closed-minded, right? But they each serve their own role. The mature dendritic cells have proven themselves in order to help present an appropriate immune response, or at least we believe that they deserve to be there. And by expressing their clout or their CD40, CD80, and CD86, these receptors then tell the killer cell or the T cells what they need to. It does a better job at convincing them what to go after. Anyways, I'm going to continue. Anyways, expression of both MCA class 1 and 2, and remember those are the major histocompatibility complex. Expression of both those classes occurs within the interstitial tissue of the testes, including the macrophages and Leydig cells. Our own unpublished results show that also testicular dendritic cells express major histocompatibility complex 2 molecules. In contrast, on the developing germ cells, MHC antigens are reduced or absent. These data indicate that spermatogenic cells are able to avoid direct recognition by the CD4 and the CD8 positive T cells, which may be important for reducing the potential for antigen-specific immune response elicited by dendritic cells or macrophages in the seminiferous epithelium. So let me... So I will continue. Interestingly, ejaculated human spermatozoa express major histocompatibility complex 1 and 2 class antigen, and the messenger RNA for human leukocyte antigen have also been demonstrated in human ejaculated spermatozoa. The principal cells in the afferent ducts may act as APCs. I forgot what APCs are. 1 second. Search APCs. Oh, antigen-presenting cells. Okay. So the principal cells in the afferent ducts may act as antigen-presenting cells since they start to express major histocompatibility complex 2 antigen after immunization with sperm antigens in rats and mice. As the afferent duct epithelium does not express major histocompatibility complex 2 under normal conditions, this finding suggests that regulation of MHC2 antigen expression in these cells may be one mechanism that protects the sperm from an autoimmune attack at this site. Okay, interesting. I think I'll stop at... Yeah, I'll stop at the end of this page. Yeah, stop at the end of this page because it looks like there are only 2 more pages after that. 3. We'll see where we go. In general, antigen-presenting cells are not antigen-specific and present a wide range of different antigens, including the so-called danger signals. So antigen presenting cells, they don't just show one antigen. They don't just show one thing. They show a lot of things, right? So since they show a lot of things, one of the other things that they can present is the so-called danger signals. So the danger model proposes that stressed or damaged cells and tissues express and release heat shock proteins during injury caused by trauma, inflammation, pathogens, or toxins. And after sensing these danger cells, dendritic cells undergo profound morphological and physiological changes and begin to migrate from the periphery to the lymph nodes. This maturation includes a switch from antigen uptake to antigen presentation mode by signals specifically generated in the inflamed tissue. So what is this telling me? This is telling me that the dendritic cells are normally existing in the tissues, all happy, and they are randomly grabbing things. But when it receives a signal, and when that signal is a heat shock protein that is then will on the just trying to see if it might be expressed on the surface of the cells or it might be actually secreted into the environment. I can't remember exactly which one. But anyways, the dendritic cells sense these heat shock proteins, and when they feel these heat shock proteins, that tells them, oh, things are going down right now. I need to go into the lymph node and mature. So what I would assume is whatever those dendritic cells were recently taking up, that is probably going to be one of the most presented antigens on their surfaces. Now, I don't know this. That's coming. I'm spitballing right now, but that's what I'm understanding. Anyways, this maturation includes a switch from antigen uptake into antigen presentation mode by signals specifically generated in inflamed tissue. The activation state of the dendritic cell is crucial in determining the outcome of antigenic challenge. For example, the development of either T cell immunity or tolerance. So this is how the dendritic cells know whether or not to present antibodies that will, or not antibodies, to present antigens on their surface that will tell the T cells not to attack or tell the T cells to attack. Resting and activated dendritic cells show marked differences in their expression of various co-stimulatory molecules. Activated dendritic cells are potent stimulators of the immune response, an ability that is linked to their high expression of several co-stimulatory molecules. In contrast, the resting dendritic cells have been implicated in the generation of self-tolerance, presumably due to their reduced co-stimulatory capacity. Interesting. So what is this telling me? This tells me that dendritic cells, when they are possibly young or normal, immature, resting, they are grabbing antigens around and then they are representing them. But since they're not activated, the immune system sees these antigens on the dendritic cells and says, okay, this is normal. I'm supposed to be seeing these antigens. So I won't attack them because, for instance, I don't know exactly how it works, but maybe a immune cell, a dendritic cell, picks up an antigen from a very beneficial bacterium, right? And so it picks it up and then presents that antigens, the antigens that the bacteria had, it presents them on its surface. But so when a T cell comes by and sees these antigens on the surface of this dendritic cell, then it says, oh, this is normal. This is a normal bacteria, and that is beneficial for the body, and it's not harming anyone, so I might as well let it stay, right? So it sees that, and so when that cell ends up seeing the same bacterium later, thinks, okay, everything's fine. But if the same bacteria, it could be that the same bacteria or a different one starts duplicating too quickly and it starts infecting the tissue, then that is going to cause an inflammatory response. And then once that inflammatory response happens, then that tells the dendritic cells you need to mature. You need to bring in the how to phrase that. You need to activate in some way, and you need to tell the T cells and B cells what to attack, right? Um, anyways, okay, so and then okay, they changed. They're on a different subject for me, but so when that happens, they go in, they mature, and then after they mature, then they can present those same antigens to the T cells and tell them what to attack. That makes sense to me. Anyways, presentation of self-antigens, or the antigens that your own body produces, by dendritic cells is likely to play an important role in the initiation of autoimmunity and its progression towards clinically important autoimmune diseases. So what does this tell me? What does this tell me? So if the dendritic cell screws up and it somehow starts presenting antigens that your body produces, but it does it in a way that warns T cells to attack, that is implicated in the start of a lot of autoimmune diseases. So what can this tell me as well for what does this tell me for SARS-CoV-2? Let's say you have a spike protein floating around, and then this dendritic cell uptakes it because it's an antigen uptaking organism, creature, thing, or immune cell, and then it takes that spike, it then breaks it up, and then presents parts of the spike protein on its surface. If some of those spikes, or some of those portions, mimic or look similar to human proteins or peptides, then it could be, unfortunately, that you end up causing the stimulation of T cells that are producing the antibodies against those antigens. And since they cross over, there's cross-reactivity with human peptides and proteins, you then end up getting an autoimmune response as a result. So this is implicated in SARS-CoV-2 and influenza for the ACTH, or the corticotropin protein, peptide hormone. And when this happens, then your body ends up creating autoantibodies against corticotropin, and then those antibodies will sequester the corticotropin while leaving the virus, giving the virus more time to duplicate, because the antibodies are being consumed by corticotropin. So the viruses duplicate a little bit longer, and what happens as a result is your immune response gets dulled, or I should say, not dulled. When that happens, you end up having a hyperinflamed response, which ends up making you very symptomatic, because by inhibiting corticotropin, you are inhibiting the release of cortisol. And when you inhibit the release of cortisol, you are creating a very pro-inflammatory environment, right? So this is how I shouldn't say SARS, but coronaviruses and influenza, how they are able to create these pro-inflammatory responses, which then result in a very symptomatic progression. And oftentimes, symptomatic progression helps aid the virus to spread, because when you are symptomatic, you end up being more juicy and leakier and sneezier and more expressive. So by doing that, a virus is more able to spread into its next victim. Anyways, on a tangent, but we can keep on going. Dendritic cells presenting autoantigens cause organ-specific T-cell mediated autoimmune diseases such as type 1 diabetes, autoimmune myocarditis, and multiple sclerosis and rheumatoid arthritis. Abnormal co-stimulatory phenotype and function of dendritic cells has been shown to in severe murine lupus. Murine lupus. Interesting. Remember, murine means mouse. So the abnormal co-stimulatory phenotype and function of dendritic cells has been shown in severe murine lupus. So furthermore, the functional phenotype of dendritic cells purified from the central nervous system of mice with experimental autoimmune encephalomyelitis is strikingly different from that of other dendritic cell populations. They are unable to prime naive T cells. Let me read this for a little bit. Furthermore, the functional phenotype. Okay. And I believe the priming of the T cells is when they are told what antigen they're supposed to attack. Let me look this up. There is a really good anime called Cells at Work. Oh no. Okay. For some reason, my computer sucks. So a naive T cell among these are the naive forms of helper T cells and cytotoxic T cells. A naive T cell is considered immature and unlike activated memory T cells and has not encountered its cognate antigen within the periphery. So yeah, this is one that has not learned what antigen it's supposed to attack. Right. So furthermore, the functional phenotypes of dendritic cells purified from the central nervous system of mice with experimental autoimmune encephalomyelitis is strikingly different from other dendritic cell populations. They are unable to prime naive T cells. They're unable to teach the T cells what to attack. So I'm trying to understand why they left this in here, but if you understand it, leave it in the comments. I'm having a brain fart right now when it comes to this. So continuing, in spite of their potential importance in maintaining the balance of the testicular immune status between tolerant and autoimmunogenic states, the dendritic cells in the male gonad have received little attention in the past. Cells that express dendritic cell markers or possess dendritic cell morphology have been observed in the testes of mice, rats, and humans. However, as the markers used are also found on macrophages, a reliable identification is difficult. So markers could be different types of proteins that are expressed, and so when it comes to different cells, we can figure out what cell it is based on what proteins it's expressing. But there is some overlap between what proteins different immune cells express. So sometimes it is difficult to do a positive identification of an immune cell. Okay, continuing. In a recent study, the presence of dendritic cells in normal and chronically inflamed testes from Y-star and these particular rats was determined and quantified for the first time using the dendritic cell specific markers. So I should say they used to have a big problem, but then they figured out better dendritic cell specific markers. And in experimentally induced, experimentally autoimmune orchiditis or orchiditis, man, EAO, this is the inflammation of the balls that are experimentally generated. DCs, and this is specifically the autoimmune one, so they find a way to cause an autoimmune attack on the testes experimentally. The dendritic cells were found in the interstitial space of the testis and in large numbers. So whenever you do get the inflammatory response caused by the experimental autoimmune orchiditis, it ends up, orchitis, I should say, it ends up increasing the numbers of dendritic cells in the interstitial space. Interesting. And in large numbers in the granulomas. And although increases of between 5.5-fold and 8-fold were seen compared with controls, these quantities are still significantly lower than the number of macrophages found in similar circumstances. So even if you do have an increase in dendritic cells, it seems to be less of an increase than in other parts of the body, which goes to show you there must still be a protective role of some form in the testes. These people demonstrated that the co-stimulatory molecules CD80 and CD86 are not expressed in the testis of normal BALB/C mice and 4-week-old non-obese diabetic mice, a rodent model of autoimmune diseases. So these are model mice that are used for specific experiments. As the absence of co-stimulatory molecules on antigen-presenting cells during antigen presentation has been shown to induce clonal energy and consequently peripheral tolerance in other systems. Let me read that again. So the absence of these molecules are normally associated with tolerance or the suppression of the immune system at certain sites. So why are these rodent models for autoimmune diseases? I would assume that they would normally be having or less tolerogenicity. Maybe they use them as rodent models for autoimmune diseases in order to induce them in some way. Okay, regardless, we'll keep on going. This finding would suggest that local activation of T lymphocytes is not possible in the normal testis. However, C80 and C86 were found in the 14 to 22-week-old non-obese diabetic mice, and our own data clearly showed the detection of the CD80 and CD86 positive cells in rat testes. This suggests that the DC-dependent triggering of activation of naive T lymphocytes by binding to the specific antigens is at the very least possible in the rodent testes. So they're saying that it is possible that the dendritic cells of the testes could be playing a role. Interesting. Okay. In light of the danger model, the recent characteristic of numerous heat shock proteins as testicular autoantigens could provide the mechanism for how dendritic cells in the testes participate in the activation of autoreactive lymphocytes and in the subsequent damage of testicular tissue, thereby overcoming the immune privilege. So dendritic cells could be implicated in the ability for your immune system to override the immune privilege, which could manifest itself in certain diseases or certain symptomology, pathology. Anyways, provided evidence that heat shock protein 70, when released by necrotic cells, acts like a danger signal by enhancing the maturation of dendritic cells, which then trigger autoimmunity. Interestingly, heat shock protein 70 did not upregulate co-stimulatory molecules on dendritic cells, but helped antigen-presenting cells become better stimulators of T cells. Although the exact mechanism remains elusive, it is important to note that the release of endogenous inflammatory signals requires necrotic cell death, such as that resulting from infection or injury. Based on our own data and that of other autoimmune disease models, we hypothesize that immature dendritic cells, normally involved in maintaining immune privilege, under inflammatory pathological conditions, sense self-antigens such as heat shock protein 70 as danger signals, and after maturation may overcome immune privilege and immune tolerance by local activation and expansion of autoreactive T cells. A A recent study by these people on human multiple sclerosis revealed that despite overcoming the immune privilege in the brain, most dendritic cells maintained an immature phenotype, and that few that did acquire maturity were most likely under the influence of inflammatory cytokines and or direct interaction with T cells. So, what they're saying here is that these dendritic cells are normally presenting antigens, but doing it in a way that suppresses the immune response because things are going good. So therefore, the antigens that you're seeing right now are good antigens that don't cause any problem. But there could be a situation such as the release of heat shock protein 70, which would be the result of dying cells nearby. If a dendritic cell experiences that signal, then that dendritic cell, with the antigens that it presents, then says, oh crap, I need to go mature. I'm undergoing a trauma, and therefore I need to mature quickly. That is basically what's happening to these dendritic cells. And then when that trauma happens to these poor dendritic cells, the things that actually kind of sad, the things that it thought that was normal in its childhood, the things that it was normally presenting, the things that it was considering good, it takes those things and then matures in a way that says these things are actually bad. These things are actually things that we need to attack. And that would normally be good, right? That would normally be good for your immune system, where it's the dendritic cells are, let's say you're not in immune privileged spots. Normally, the dendritic cells, immature, taking up new antigens at all times, and then if things get really bad, like such as a heat shock protein or necrosis is happening nearby, then they mature, and then they start the antigens they present are used by the other immune cells and say, these are bad antigens, therefore I need to attack those bad antigens. That makes sense. But what would be bad is if this happens in an immune privileged site, such as the testes. And then those dendritic cells that were probably playing a very large role in tolerogenicity or suppressing the immune response, they might end up becoming pro-inflammatory and pro-autoimmune response if they do get exposed to tumor necrosis factors, which would, therefore, what this says to me is, don't allow trauma to happen to your balls. Don't allow trauma to happen to your eye. Don't allow trauma to happen to your central nervous system, because with that increase in trauma, you are then you are convincing the dendritic cells to not be immunosuppressive. You're telling those dendritic cells to be mature and to initiate or convince the cells around it to attack the antigens. Now, normally it's not a big issue, but if it's the dendritic cells of these immune privileged sites, we need to be very mindful that if they get traumatized, then the antigens that they are presenting were supposed to be suppressed, or were supposed to convince the immune cells to not attack. Now they are saying, oh, I should attack those. And that can cause more trauma. That could be a positive feedback loop, which could then permanently cause chronic issues. So now I'm thinking, what about the trauma that football players, encephalitis, I think? Well, if you have a lot of trauma to the brain, multiple concussions, that could be causing cell death. And by causing cell death, they are releasing their heat shock proteins and they're releasing necrotic factors, right? And when that happens, then the dendritic cells are have the unfortunately, go from being naive, go from not naive, but go from being immature and immunosuppressive, and then they become immunoreactive. And when that happens, then you might start creating antibodies, which end up damaging proteins that are normally supposed to be protected. So that might be one of the contributing factors to uh, the the brain degeneration of football players. I'll I'll have to look into that. There's probably a paper about that. Anyways, I'll keep on going. Interestingly, increased levels of tumor necrosis factor alpha were found in Orchitis testis, and tumor necrosis factor alpha is one of the several agents that induces the phenotypic changes characteristic of mature dendritic cells, and can currently act as a regulatory factor of dendritic cell migration in inflamed tissues. Interesting. So here they have an image of the hypothetical model of the impairment of testicular immune privilege in the testicular inflammation involving dendritic cells. So here we have our immature dendritic cell, and here they're CD880, they're CD886, they're CD40, and they're high intracellular major histocompatibility complex 2. These are all low, and these are all high, and so you have an immature dendritic cell. And so whatever antigens they're taking and presenting, that is telling the immune system not to attack these antigens. Do not attack them because they're good. Things are going great. Do not attack these antigens. But if you have germ cell loss or necrosis, or if bad things happen, let's say you got kicked in the balls and it was a pretty hard kick and it ends up damaging the tissue significantly, then these dead cells are releasing these PAMP, the heat shock protein 70, and autoantigens. So these autoantigens, they might be taken up, but they might not do anything. Normally, they might have actually had a protective role for the dendritic cells, normally, but because of the PAMP and the heat shock protein 70, then they end up going from an immature dendritic cell to a mature dendritic cell, right? And so when it's the mature dendritic cell, they have high CD880, high CD86, and high CD40, and then this is on the surface of them, which means when a T cell ends up interacting with the mature dendritic cell, and this mature dendritic cell is expressing some of these autoantigens, it then tells the T cell, oh, you need to attack this antigen, which can then cause autoimmune responses because these are autoantigens. These are antigens that your own body produces, and it's not good, or we should say, if it's dysregulated, it is not good for you to produce antibodies against yourself. Interesting. So anyways, the hypothetical model of the impairment of testicular immune privilege in testicular inflammation involving dendritic cells. So in chronic testicular inflammation, the number of dendritic cells in the testis is greatly increased as a consequence of a temporary or silent infection. Necrosis of testicular cells can lead to the result of self-antigens. Well, this is another thing that that I didn't even consider, but now I might, where I'm worried, since orchitis has been found in the autopsies of people with COVID-19, and it wouldn't even need to be the virus itself infecting the testes. We just know that orchitis is happening in a large portion of people who die with um, with COVID-19. I have not seen any studies talking about orchitis. Sorry, there was one. There was one instance of orchitis in a child in Brazil. I saw that. I don't know if it was published yet, but it is happening in people infected with COVID-19. So if this happens in someone with COVID-19, maybe they have a mild case, right? Maybe they don't even notice the symptoms, right? But what if they get some minor inflammation in their testes, and then that minor inflammation ends up eliciting an immune response, right? Then what? And we know that chronic inflammation of the testis is hard to diagnose medically because people don't know. People don't even know that their balls are inflamed. Um, People don't even know to go into the doctor for it. So if we are presented with this virus that we know can cause inflammation of the testis, and we do know that this virus, uh, if presented the opportunity, would be able to infect the cells of the testis. And I should also mention that the germ cells, these germ cells here, are the highest expressing cells in the human body of the ACE2 receptor. So they are the most, germ cells are the most ACE2 positive cells in the human body, right? So if a virus was able to get into a germ cell and kill it, rupture it, induce inflammation, and not just that germ cell, it'd probably be able to get into multiple germ cells. Um, this worries me because then we might be initiating this entire sequence of events where an immature dendritic cell ends up getting some of the autoantigens from the dying germ cells and then then matures into a mature dendritic cell and then will permanently or semi-permanently create an autoimmune response for the testes. We might start seeing some very serious infertility issues within the next few years. Which is very unfortunate, but um, that might be the case. So my hope that whatever this is, it is temporary. Um, and whatever if we don't get this pandemic under control, I do have a worry that there will be a massive reduction in the human population in the next over the next few decades. I really hope I'm wrong. I really, really hope I'm wrong. I know that some people might cheer it on, but the human population might seriously be hurt from this virus. Um, But, some people will argue, we were overpopulated anyway. But we were making good strides in of ourselves to control our own population uh, consensually. Right? We were having more, um, what would the word be? Birth. We had more birth control. We were increasing the standard of living for so many people. We were stopping a lot of pediatric disorders, um, which then allowed families to not have to worry to make as many children to survive. They could have fewer children, and they knew that those few children were much more likely to survive, and then they could invest more resources into those fewer children. Um, we were making excellent strides in family unit control, and we didn't we don't need we didn't need to have a traumatic event lower the world's population. We don't need a traumatic autoimmune response to reduce the human population. We were making strides to do that in of our own volition. We were really doing a good job at knowing and learning um, our limits, at learning our limits. Just have so much fear about this because there might be a lot of there 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 could be a lot of children that wanted to grow up with families, and if they get infected with this, is this going to permanently affect their ability to reproduce? Um, Maybe not children, but maybe post-pubescent teenagers. Maybe their dream was to start a family and raise children, and what if they can't do that because we as a society just said, yeah, everyone's going to get sick anyway, so let it take over us. It's fucking bullshit. How dare we? How dare we like allow a virus like this to spread? We should do everything, absolutely everything in our power to make sure an event like this doesn't happen to our body and to the bodies of everyone else. This affects children too. This affects young people. Sure, old people are dying disproportionately, but that doesn't mean that all of these other effects are also things that we should consider. I I think I'm going to stop the discussion here, um, because I think we've gone into a pretty heavy area, and it looks like, yeah, I'll stop here. I'll do another video on this. There are some other things that other videos that I want to put out first, so I will continue this review later. But what I'm gonna switch to now is the the discussion on autoimmunity and why. God dang it! Why do we need to be careful about diseases? Well, we don't know all of the effects that a disease can do to you. And let's go to the Spanish influenza. Let me look at that. Let me see if was that actually an influenza or was it Spanish influenza? Yeah, so that was a H1N1 virus. So with the Spanish flu, that was a disease that mammals and other animals have been has had a relationship with. Influenza viruses we've had a relationship with for a very long time. And so the people who exist and live now, the animals that exist and live now, very frequently we have mechanisms to deal with those diseases because we've co-evolved with them for a significant period of time. So if people contract influenza, we know what happens. Like to a large degree, we have an understanding of the types of symptoms you can get. What is the disease progression in more serious cases? What is the disease progression in mild cases? We have we have co-evolved with influenzas for a very long time. So whatever an influenza strain can throw at your body, we assume that the mechanisms, the cellular signaling in your body, has at least some recognition of it and knows to a degree what to do, and it's not going to throw it off kilter as much. This disease, SARS-CoV-2, we have absolutely no clue what it's going to do. No clue at all. Yes, we have 4 other distantly related coronaviruses, but we have no clue what this one's going to do. This is the first highly pathogenic coronavirus of the modern era. We don't know what it's going to do. The other coronaviruses, the human coronaviruses, I should say, so not MERS and not SARS-1, they're benign. They hardly do anything to humans. They infect us multiple times throughout our lifetime, but they're benign. And as far as I know, they don't cause orchitis. They don't cause inflammation of the balls. The more pathogenic strains, SARS-1, MERS, SARS-CoV-2, they do cause orchitis. They do cause inflammation of the balls. And what does this mean for us? I don't know. With the other, I don't know if there was a study done on the initial SARS on what happened with their fertility, right? Could have affected it negatively, but what was it? Less than 8,000 people got it, so it's hard to make generalizations on a cohort that's small. It's hard to find samples, but yeah, right now there are so many people being genetically engineered with. I mean, I'm using genetic engineering in its most liberal sense because viruses do genetic engineering, right? So I don't know. All I know is that it's fitting a pattern, and if we fit the pattern with these other diseases similar to it, it's not looking good. And I would invite, I'm going to drink, I would invite whoever's listening to present some questions and some counter arguments, because I'm really getting into my own head about this, where I am worried. I am incredibly worried about what is going to happen to society when we let this sweep over all of us and affect all of our cellular signaling. It's been a while. It's been half a year since all of this started, right? So we would hope that if there were issues earlier, or if there's some long-term issues, then we would start seeing those long-term issues soon in the literature or in the clinical studies of these people. Because, I mean, we've gone through 6 months. So 6 months ago, we had, a few dozen cases, right? So we just need more time, a few more months, and then we can start generating data on what are the semi-long term consequences of contracting coronavirus. And then after that happens, then people will collect the data and then they will write something and then probably put it on a preprint server, and then we can analyze it and learn a little bit more. But what I cannot fathom is why this isn't even being talked about in the media. Why is the testicular vulnerability of this coronavirus not being discussed in the media? It makes no sense. This is something that if you told people, hey, your balls might be at risk. If this was well known and well distributed, then you might see a better, more proactive response among the populace. Because a lot of men will do a lot to protect their balls. I mean, men will do a lot to protect their testosterone production. Men will do a lot to control their prolactin production. And yet, that is not talked about at all. And there were plenty of papers, like at the very beginning, saying you gotta worry about this because this happened with SARS and MERS. The testicular inflammation, it's something you need to worry about, right? But isn't there? I'm speaking to the void, but isn't there a there's a science fiction novel where this guy ended up, he was like one of the last fertile men in the world. Like, I obviously don't think it's going to get to that point where there's only a handful of fertile men left in the world. But what I do think, oh, and yeah, it stopped because it's reached 2 hours. So what I do think is that there will be a significant number of people who wanted to have children, um, who won't be able to, and it could have been avoided. And I'm also worried that there's going to be a significant number of people who have long-term hormonal changes that, had they known they would have to live with the long-term hormonal changes, they wouldn't have consented to contracting the disease, right? So, what I guess what I am presenting, what I am worried about, is where does society go after this? What, how will society react if we find out that this does cause long-term sexual health repercussions? What do we do as society when our population, if our population plummets from our inability to reproduce? I really don't know. 1 second, the cat wants out. Do you want out, Walter? Traded one cat for another. So, what, I present this to you. What do you think is going to happen? Because right now I'm about 40% sure that that is a going to be a consequence of all this, is a significant reduction in fertility and a significant alteration in population-wide hormone levels. It's almost as if, gosh. And then that will also result in population-wide behavioral changes, because we do know that hormone levels are associated with your psychology. They're associated with your ability to think. They're associated with everything. So, yeah, that is all I want to mention about this. What I guess I will. What else do you want to talk about? I don't think anything. I think I'm going to leave it at this, and I'm going to just enjoy the rest of my day, because all this is getting me a little sad. So I brought this up to warn about the problems associated with long-term transmissibility of the virus if it does get a hold in the testes. And what I'm seeing now is that there are instances of viral RNA present in the semen, and then also the presence of inflammation of the testicles. And I have seen the presence of heavily increased prolactin levels and luteinizing hormone levels, and decreases in the testosterone to luteinizing hormone ratios, and yeah, there is a problem going on right now, and I really hope that this research starts being published soon, because the lack of information that is being presented in the literature is really frustrating me. it really is frustrating me how I don't have any information uh, about the testicles in coronavirus patients. So with that, I'm going to leave because I am going to relax the rest of the day, and I hope that you all four of my subscribers, have a good evening as well. So stay safe, stay healthy, and uh, uh, yeah, just stay safe and stay healthy.